Hey there, this is Dr. Rosario. Remember in our last lecture, I said that the most common form of chemical potential energy in living things is the sugar glucose. Cells convert the energy stored in glucose into a usable form, ATP. In the next few videos, we are going to talk about the different pathways that cells use to extract energy from that molecule of glucose. In this video, we are going to focus on a pathway called aerobic respiration. In the next video, we will talk about some of the other pathways that organisms can use. These are incredibly complicated biochemical processes with lots of unfamiliar terminology. I don't want you to get lost in the details, but you do need to understand some of the basic concepts. This is important for people interested in healthcare for two reasons. One is that you need to know how cells get energy to grow so that you can control microbial growth. And second, we often use biochemical characteristics of microorganisms to identify them in the laboratory. As you study this material, make sure to use these learning goals to help you identify what you truly need to know for this class. Glucose can be completely broken down to carbon dioxide in a single step. This is what happens when we burn a wooden log. Wood is primarily made of chains of glucose and the polysaccharide cellulose. In the combustion or burning of glucose, electrons are stripped from the glucose molecules and given to oxygen molecules in the air. This is a redox reaction, like the ones that we talked about in the last video lecture. As glucose is oxidized, it loses electrons. Bonds are broken, carbon dioxide and energy are released. Oxygen, meanwhile, acts as an electron acceptor, gaining those electrons to form water molecules. While this process is useful if you want to stay warm on a cold night on the beach, it isn't particularly useful to cells. Most of the energy in combustion is released as heat. For cells to capture that energy and put it into a form that can be used for cellular work, namely ATP, glucose breakdown or catabolism occurs in a series of enzymatically controlled reactions. We are going to learn about three different pathways that microbial cells use to extract energy from glucose. All three pathways start out the same, but diverge in terms of their products and the amount of energy that they release. In this video, we will focus our attention on aerobic respiration. In the next video, we will look at fermentation and anaerobic respiration. As you can probably figure out from the name, aerobic respiration requires oxygen. Fermentation and anaerobic respiration do not. Aerobic respiration is like the combustion of wood. It involves the complete oxidation of glucose to carbon dioxide and the reduction of molecular oxygen to water. Right? These two things have to be paired together. Instead of a single step, however, glucose oxidation in cells is done in a stepwise fashion. This allows the cells to systematically extract 36 to 38 ATP per molecule of glucose, and that's a lot of energy. We're not going to go into deep detail here, but instead we're going to focus on the major steps of aerobic respiration. We're going to divide the process in two: the oxidation of glucose and then the reduction of oxygen. In the last video, I talked about the importance of electron carriers. NAD and another electron carrier, FAD, play a very important role in pairing glucose oxidation with oxygen reduction during aerobic respiration. First, electrons are removed from glucose and given to the electron carriers during glucose oxidation. Eventually, those same electrons are passed to molecular oxygen during oxygen reduction. By the end of this video, you should be able to see how these two processes come together to help make ATP for the cell. We will start by looking at glucose oxidation. During this process, one molecule of glucose is broken down into 6 molecules of carbon dioxide. Glucose has 6 carbon atoms. Each one of those carbon dioxide molecules has 1 carbon atom, so the carbon balances out by the end. In aerobic respiration, there are 3 stages of glucose oxidation: glycolysis, the intermediate step, and the Krebs cycle. So you should know those 3 stages of glucose oxidation. In prokaryotic cells, all 3 of these stages occur in the cytoplasm of the cell. In eukaryotes, glycolysis occurs in the cytoplasm, but the intermediate step and the Krebs cycle occur in the mitochondria. But the processes are essentially the same. As we progress through these stages, the bonds that hold those 6 carbon atoms together in glucose are broken. 2 molecules of carbon dioxide are released in the intermediate step, and 4 molecules of carbon dioxide are released in the Krebs cycle. Most of the usable energy from the glucose molecule is extracted in the form of high-energy electrons. These electrons are removed from glucose and given to the electron carrier molecules NAD and FAD. This form of energy is not immediately usable by the cell, but will need to be converted to ATP. This will happen eventually when NADH and FADH2, the reduced form of these electron carriers, will transfer their electrons to oxygen in the second half of aerobic respiration. A little bit of ATP, which is immediately available for cellular work, is produced during glycolysis and the Krebs cycle. The production of ATP during these 2 stages is called substrate level phosphorylation. Right? That's an important term to remember, substrate level phosphorylation. Now remember, to make ATP, we must add a phosphate group to adenosine diphosphate, or ADP. During glycolysis and in the Krebs cycle, some of the energy of glucose oxidation is used directly to attach phosphate groups to organic molecules. These phosphates are then transferred to ADP by enzymes called kinases. So that's what you're seeing illustrated on this slide. The organic molecule and ADP are both enzyme substrates, which is why we call this process substrate level phosphorylation. So later we will talk about oxidative phosphorylation, which is another way that cells make ATP, and that's going to occur as the result of oxygen reduction. So here in glycolysis in the Krebs cycle, we have substrate level phosphorylation, where phosphate groups are transferred from an organic molecule to ADP to make ATP. That's going to be mediated by an enzyme where the organic molecule and the ADP are both substrates. So this is called substrate level phosphorylation. At this point, you should go ahead and stop this video and make a note of what goes into each stage of glucose oxidation and what comes out. What are the inputs? What are the outputs? What are the reactants? What are the products? As you read through the textbook, you will notice that glucose oxidation is a complex process. There are many steps and many complicated chemical names. At a minimum for this class, you are responsible for knowing the information that is in this figure. Not just the names of the molecules, but the numbers as well. So go ahead and pause the video. I'll give you time. Okay, so now at this point, where is the energy that was stored in that glucose molecule? Some of the energy has been converted to ATP, right, during glycolysis and during the Krebs cycle. That energy can immediately be used by the cell, and the remainder of the energy can be found in those high energy electrons carried by NADH and FADH2. In aerobic respiration, remember this is going to involve the reduction of oxygen, the electrons found on NADH and FADH2 are carried to a series of proteins called an electron transport chain. These proteins are embedded within a membrane. In prokaryotes, this is the plasma membrane, and in eukaryotes, it is the inner mitochondrial membrane. The process that occurs within these membranes is essentially the same whether we're talking about prokaryotes or eukaryotes. The electrons are passed from one molecule to the next until they reach a molecule of oxygen or oxygen gas, O2. The oxygen atoms then combine with hydrogen atoms to form water. Meanwhile, as the electrons traveled through the electron transport chain, the energy that was associated with those electrons is used to move hydrogen ions out of the cell or out of the mitochondrial matrix. This creates a difference in concentration of hydrogen ions across the membrane. This difference is called an electrochemical gradient. The energy that started out on glucose has now undergone another transformation. It started out here on NADH and FADH2, and then it moved from there to the hydrogen ions in this gradient, this electrochemical gradient. The energy that is stored in the hydrogen ion gradient is captured as the ions flow down the gradient through a protein called the ATP synthase. This transmembrane protein, it spans the membrane, has two main components: a channel that allows for the movement of hydrogen ions and an enzymatic portion that combines ADP with inorganic phosphate to make ATP. As the hydrogen ions diffuse down their gradient through this channel, it powers the molecular machinery that creates the high-energy phosphate bonds. This is oxidative phosphorylation. So the energy that is stored within the hydrogen ion gradient is used to phosphorylate ADP. For every glucose molecule that is oxidized through glycolysis, the intermediate step, and the Krebs cycle, 32 to 34 ATP are produced through oxidative phosphorylation. This is substantially more than is made through glycolysis in the Krebs cycle. Remember, you had 2 ATP that were made in glycolysis and 2 ATP that were made in the Krebs cycle. Again, remember that was substrate level phosphorylation. So here you can see you get a whole lot more ATP through the second half of this process, through the reduction of oxygen and the building of this hydrogen ion gradient. The proteins that make up the electron transport chain are not the same in all microorganisms. In some, but not all organisms that use oxygen, the final protein in the chain is an enzyme called cytochrome C oxidase. So it's this final protein, and it's called cytochrome C oxidase. This protein passes electrons from a molecule called cytochrome C to molecular oxygen to produce water. So it's oxidizing cytochrome C, right? That's why it has that name. Now we can use this information, knowing that some organisms have this enzyme but others don't, as a way to characterize microorganisms in the laboratory, to differentiate between different species of bacteria. And there is a relatively easy assay called the oxidase test to identify organisms that have the enzyme. So there is a chemical that we can add to cells that will turn dark blue or purple in the presence of the enzyme. So you can see in the image on the right, you can see that the first swab has bacteria on it that are oxidase negative, and the one on the right has bacteria on it that are oxidase positive. So this can be especially useful when we're characterizing organisms that are gram-negative rods. There is a large family of gram-negative rods called the Enterobacteriaceae, and this family has lots of notorious human pathogens in it, like Salmonella, Shigella, E. coli, and even the organism that causes the plague, Yersinia pestis. They're all part of this big family. And all of our Enterobacteriaceae are oxidase negative. So if we have a gram-negative rod and we're not sure if it's part of that family or not, we can do the oxidase test and we can eliminate a whole group of microorganisms if it's oxidase positive. We can say, oh, nope, it's not Enterobacteriaceae. And this is just one biochemical tool that can be used to differentiate between species of bacteria that is based on their metabolism, based on their ability to respire or to not respire. In this video, we've talked about just one of the ways that microorganisms obtain energy from their food. Here we have focused only on glucose as the food source, and aerobic respiration is the most efficient way to extract energy from glucose. In the next video, we're going to talk about some of the alternatives to aerobic respiration and talk about some of the ways that microorganisms can get energy from other foods besides glucose, other molecules besides glucose.